In this lecture, we're going to discuss replication and cell division. At the end of this lecture, you should be able to identify the enzymes required to replicate DNA. You should be able to define numerous terms that we'll be using in this lecture regarding their role in DNA duplication. You should explain the steps involved in DNA replication, mitosis, and meiosis, and also compare and contrast bacterial cell division from eukaryotic cell division. In our cells, DNA is supercoiled. Supercoiling is a term which just means that the DNA is twisted and wrapped up into a little ball. So if you were to take a rubber band and twist it from both ends until it just becomes this mass, this very compact mass, that's what supercoiling is. And DNA supercoils or twists up in a specific direction. So DNA is negatively supercoiled. So this negative supercoiling is what is going to allow replication and transcription to occur, because that negative supercoiling doesn't require as much energy to separate the double helix. And we need to separate the double helix in order to replicate DNA. In eukaryotic cells, those negative supercoils are maintained by the DNA double helix being wrapped around histones. DNA is duplicated during replication. So you have your mother cell with DNA in it. That DNA is duplicated so that you end up with two identical copies, two cells that are identical to that mother cell. So during cell division, each of these identical cells or daughter cells gets a complete copy of the genome from the mother cell. Replication always begins at a specific spot in the genome, and this spot is the origin of replication, or ORI. In bacteria, there's only one origin of replication. So replication occurs at one specific spot in that bacterial genome, and then it continues until the entire circular genome is copied. In eukaryotes, however, there are numerous origins of replication throughout the entire genome. So you're going to have simultaneous replication going on in different areas of the genome. Replication is considered to be semi-conservative because each progeny cell has exactly half of the original DNA molecule. So one half of that DNA molecule, or one strand, is from the original cell. The other strand is what's the replicated, what's made. So that's why it's called semi-conservative. One of those daughter cells has the right strand from the mother cell, and the other daughter strand has that original left strand. So this depiction of semi-conservative replication, where you have the original parental double-stranded DNA. That double-stranded DNA has to break apart. Each daughter cell gets one half of that original double-stranded DNA, and another strand is then made so that you'd get two double-stranded pieces of DNA. So we already said that the double-stranded DNA has to separate in order for it to be replicated. So we already know that the DNA is held together through hydrogen bonding, and also the helix is twisted around and wrapped up. The DNA, we already said, is negatively supercoiled. So both the supercoil and the double helix have to be unwound in order for that DNA to be replicated. So we already said that DNA is supercoiled, and we already know it's a double helix, and these need to be separated in order for replication to occur. So how do we unwind or separate the double helix and the supercoil? Well, we do this through the use of enzymes. So our supercoils are unwound by topoisomerases. There are several topoisomerases. One very important topoisomerase is the enzyme gyrase. Gyrase cuts both strands of the double-stranded DNA, which results in a double-strand break. Now, gyrase is also going to hold on to those cut ends so that they don't go and connect with other things or rejoin. Those two halves have to unwind, and then the ends are rejoined together. So gyrase has a very important function in replication. The double helix is unwound by the enzyme helicase. What helicase does is it disrupts those hydrogen bonds that hold those base pairs together so that those can be separated. So we have a schematic of how this unwinding occurs. So that parental DNA is a double helix, and it's supercoiled, twisted up together. So you're going to use your topoisomerases, including gyrase, and your helicase to unwind those supercoils and then also break apart that double-stranded helix. In bacteria, such as E. coli, you have one origin of replication. You have proteins that are going to come in and bind to that origin of replication. You're going to have your enzymes that are going to unwind the double-stranded DNA and so that you can replicate. So in E. coli, you have your helicase that's going to come in and unwind that double helix. You're going to have single-stranded DNA binding proteins to keep those two strands separated from one another. You're going to have RNA primers that are made to initiate replication of that strand. That replication occurs in two forks, and so each strand is being replicated at the same time. In eukaryotic replication, we already said there are several origins of replications, not only one. You get initiation of replication at each origin of replication site. You get those two forks that are going to replicate in opposite directions until the origins of replications, how they're replicating, meet together. And before you know it, the entire genome is copied. So we already mentioned single-stranded binding proteins, and those bind to the double strands that after they're separated so that they won't come back together. This concept is called parental divorce because the two parental DNA strands in the double helix have to be separated, and they cannot rebind because they're being replicated, and each strand is then going to be the template for a new strand. So these single-stranded binding proteins bind to each of those single strands and keep them apart. So here's the schematic of your double-stranded DNA that's separated. Those single-stranded binding proteins come along and bind to the single strands so that they don't come back together, because DNA wants to be double-stranded. If it has the opportunity, it is going to bind to the complementary sequence. So the single-stranded binding proteins are really important for replication. When we make that new DNA strand, we have to unwind the double helix. The enzyme primase comes in and makes RNA primer molecules. We need these primers to initiate the making of that new strand. So the primer attaches, and the molecular term for the word attach is hybridize. So those primers hybridize to the DNA at that origin of replication, and then those primers will start to replicate that strand of DNA with the matching complementary nucleotides. Of course, other enzymes are needed to replicate DNA. One important enzyme is DNA polymerase 3. DNA polymerase 3 attaches to the new DNTPs or deoxynucleotides to the 3' hydroxyl end of the leading strand, and that's going to allow elongation toward the replication fork. So polymerase 3 is going to allow replication in the 5' to 3' direction. You need more primers that are going to attach to the opposite strand to elongate that. The opposite strand is called the lagging strand. So we have our two sides of our double-stranded DNA. One side is considered the leading strand, and the other side is considered the lagging strand. The enzyme DNA polymerase 3 has two subunits. One of those subunits manufactures that new strand of DNA. The other subunit is a donut shape, and it's considered the sliding clamp. That donut shape wraps around the single-stranded DNA and moves along that single-stranded DNA. So here we have a picture of our double-stranded helical DNA that becomes separated into two single strands with the aid of our single-stranded DNA binding protein. So keep in mind, helicase is going to unwind the helix. Single-stranded DNA binding proteins are going to come in and attach to keep those two strands separated. You have one side that's the leading strand and one side that's the lagging strand. So why do we have a leading strand and a lagging strand? Remember, DNA is antiparallel. One strand is in the 5' to 3' direction. The other strand is in the 3' to 5' direction. Replication always occurs in the 5' to 3' direction. So for one of those strands, when you have the 3' end, that 5' new strand gets started and can keep going up and making new DNA. That's the leading strand. Well, the other side is opposite. That's the lagging strand. So the DNA is being replicated non-continuously. So we'll talk a little bit about how that occurs. Another enzyme that's important is DNA polymerase 1. And DNA polymerase 1 is like our whiteout. It's an editing enzyme. What it does is it attaches to nicks that might be made during replication in the growing DNA strand. And what it's going to do is it's going to edit. So if there's a little nick that's made, if there's a mistake, it's going to go in and fix that. So we have our template DNA, we have our initiation of RNA synthesis with an RNA primer, and then we have extension of that RNA primer with DNA polymerase, which we add on our nucleotides complementary. So we already said DNA is antiparallel. We have a leading strand and a lagging strand. The leading strand DNA is replicated continuously, and the lagging strand is not replicated continuously. So short segments are continuously being made, but not continuously just moving along that strand. So in the lagging strand, you have tiny pieces of newly synthesized DNA being made that are then going to be glued together. So these short pieces of newly synthesized DNA in the lagging strand are called Okazaki fragments. Once again, we have a picture, a replication fork. We have our double-stranded DNA, one strand in the 5' to 3' direction, the other strand in the 3' to 5' direction. DNA is always replicated in the 5' to 3' direction. So in the leading strand, that 5' to 3' direction can just continuously move up that fork as the DNA is separated. In the lagging strand, the DNA has to be made starting at the end of the fork and then moving down. So a little piece is made, and then another piece is made, and then another piece is made, these Okazaki fragments, until the entire strand is replicated. There are other enzymes that are very important in replication. Ligase is the enzyme that is a glue. So that's what's going to seal any nicks, seal all those Okazaki fragments together once they're all made. There are editing endonucleases that are going to replace any nucleotides that get placed in that don't match. So let's, for example, if there's a C on the parent strand and for some reason an A gets put in on the new strand, that A will be removed by endonucleases so that a G can be put in. We already mentioned gyrase is a topoisomerase, which is going to aid in unwinding those supercoils, and methylases will add methyl groups to new DNA that are going to match the original strand. So here we have our Okazaki fragments. So we have our double-stranded DNA and we have our RNA primer that's going to make a small section in the 5' to 3' direction. Another primer will be placed on and another small piece will be made, and another and another. That RNA is then going to be removed and ligase is going to glue in all that DNA so it's one continuous new strand of DNA. So another picture are topoisomerase enzymes that are going to unwind the supercoils. Our helicase, it's going to unwind our double-stranded helix. The primase that's going to come in and generate an RNA primer. Our single-stranded binding proteins that are going to hold those two steps, single-stranded DNAs apart. One strand, the leading strand, we're going to have a polymerase 3, our donut-shaped sliding clamp that's going to come through and just make another complementary strand in a continuous fashion moving up that replication fork. In the lagging strand, we're going to make small pieces of complementary DNA using these Okazaki fragments, which are then going to be glued together to make one continuous strand. So that's replication. Now we're going to move into cell division. There's bacterial cell division and eukaryotic cell division. Bacterial cell division, of course, is much simpler. So in bacteria, you have a single circular chromosome. Replication is going to proceed at the same time in both directions. You have one origin of replication. Eventually, those two replication forks meet and merge, and you end up with two exact copies of that original circular piece of DNA. So here's your picture, origin of replication. You're going to separate into two strands. You're going to get your new DNA, and they're eventually going to meet up at the other side, and you're going to end up with two identical double-stranded circular chromosomes. And these two chromosomes that are made are the daughter chromosomes. In bacterial cell division, again, you have your circular chromosome that is replicated at the one origin of replication. You end up with two double-stranded circular pieces of DNA. Your bacterial cell is going to undergo binary fission and become two cells, each with an exact copy of that original double-stranded circular genome. Eukaryotic cell division, it's a much more complicated process because there are multiple chromosomes within the cell. The genome is located inside the nucleus, which is separate from the cell. You have a membrane around that nucleus, so you have a lot more steps in cell division for eukaryotic cells. Eukaryotic chromosome is linear and very, very long. You have numerous origins of replication, so you have that replication and those replication forks going on at various points throughout the genome simultaneously. Here's a eukaryotic chromosome schematic, and you have your centromere and your kinetochore in the center of the chromosomes. For eukaryotic cell division, we have mitosis and meiosis. So first we're going to discuss mitosis. In mitosis, you get the disassembly of the nucleus of the mother cell. You have to divide those chromosomes. You have to separate the chromosomes, and the mother cell then has to divide to become two daughter cells. And then you need to rebuild that nucleus around those chromosomes that have been separated in each daughter cell. So here's your picture. You have your mother cell. You have two pieces of chromosome in the mother cell. You then get replication. You're going to make an exact copy of that DNA. Those chromosomes are then going to line up in the center of the cell, and the cell is then going to separate, and each half of the chromosome is going to be pulled apart, so you get two cells, diploid cells, that are identical to that original cell. Once again, you have homologous chromosomes. You replicate those chromosomes in replication. These replicated chromosomes are called sister chromatids. You then have mitosis, where these cells divide and you separate the sister chromatids so that you have an exact copy of the chromosome from the original parental cell into two daughter cells. The process of DNA replication takes place in the S phase or synthetic phase of mitosis, and that S phase is separated by two gap or G phases. So you have your different phases of mitosis. You have prophase, where you have chromosome condensation. You have metaphase, where the chromosomes move to the middle of the cell and align. You have anaphase, where each chromosome is then separated. And you have telophase, where you have your nuclear membrane surrounding each set of the newly divided chromosomes. So here's just your picture. You have your inner phase, which includes your gap 1 phase, your S phase, which has DNA replication, and your gap 2 phase, and then your mitosis, which you have your prophase, metaphase, anaphase, and telophase. Now in meiosis is similar to mitosis, but then you have a second division. So higher eukaryotic cells are diploid. However, our germ cells, which include our eggs and our sperm, are haploid. They only have half the number of chromosomes as a diploid cell. So instead of having 46 chromosomes, you have 23. Because you only have half of the total chromosomes in the somatic cell, they're termed haploid. So in a human egg or a sperm cell, there are 23 chromosomes. One of those chromosomes is either an X or a Y, depending on if the person is a male or female. Once you undergo fertilization, The cells in the newly formed embryo have the correct number, 46 chromosomes in the somatic cells. So fertilization restores that diploid number of 46 chromosomes. So here we go, meiosis. One diploid cell becomes 4 haploid germ cells, which are your eggs or your sperm. The chromosomes are replicated once in S phase, just like in mitosis in that S phase, into sister chromatids, but the cell divides twice. So just like mitosis, the first step is DNA replication. So you have your sister chromatids that are duplicated. During the first stage of meiosis, the homologs are segregated into different cells. So you end up with 2 cells that have 1 copy of each of the 23 chromosomes. So in the first stage of meiosis, you have crossing over. So crossing over is a very important process of meiosis. So in prophase, you have your homologous chromosomes that are going to pair up lengthwise. What crossing over is those homologous chromosomes then start swapping pieces of that chromosome back and forth, which is going to shuffle the genome, kind of like shuffling a deck of cards. You're mixing up those chromosomes. Those homologous chromosomes are then going to separate, and those new chromosomes are going to be part of the egg and sperm. So now these new chromosomes are unique because they've undergone crossing over. They've been reshuffled a bit. Then you undergo the second stage of meiosis, where those sister chromatids are pulled apart just like in mitosis, but then you have 4 cells. Each of those cells has 1 copy of the 23 chromosomes. So now they're haploid. So this whole process is what allows all of us to be unique. So here's a picture of my meiosis. You have division 1, where you have your replication and your cell division, so that you have your parental cell and that becomes your 2 daughter cells. In the second meiotic division, you have your 2 cells that then divide and become 4 haploid cells. Mitosis versus meiosis. What are the differences? In mitosis, you have 1 diploid cell that duplicates its DNA and splits that DNA up into 2 identical diploid daughter cells. In meiosis, you also have 1 diploid cell. It duplicates its DNA also in that S phase, but it splits the DNA into 4 genetically unique haploid cells. So in summary, bacteria undergo binary fission for their cellular division. Somatic cells undergo mitosis. Our germ cells, which are eggs and sperm, undergo meiosis. And we also have cytokinesis, which is the process of splitting those daughter cells apart, each getting cytoplasm, Golgi, and all of the cellular structures that each cell would need. So our cartoon, Marty, I'm just about to divide, so I can't make it for lunch, but both of us will get back to you. So in this lecture, we talked about DNA, how it's replicated in that S phase, how bacterial cells and eukaryotic cells divide, all the different enzymes that are involved in these processes. So if there's anything in this lecture that you don't understand, just shoot me an email or post a question on the discussion board.